Hello, everyone. Welcome to the second event, our Translational Neuroimaging Educational Program. In this program, we coordinate a series of presentations, discussions, and workshops with the hope to actively engage in the neuroimaging community. Our goal is to progress the translational application of computational neuroimaging technique to enhance human brain health. We do have a website for this program, so please check it out. I would like to introduce this program a little bit. This educational program is necessitated within the lifespan working group of the Enigma Consortium. By hosting the talks, presentation, and workshops, we want to promote the use of neuroimaging techniques to advance medical and scientific knowledge. This is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training background. We hope to provide opportunities to expand skills, initiate collaborations, and facilitate networking in a welcoming learning environment. The directors of this program are Dr. Shalaila, Haas, and myself, and with an international advisory committee. Please consider to join our mail list in our website if you would like to be notified when we have new updates about this program. And we do have several social media channels. Please consider to follow us. Today, we're going to have Dr. Shalaila Haas to present her latest work on using CentileBrain models to investigate the brain morphometry of individuals at clinical high risk for psychosis. And in the next session, we plan to have Lucas Roll from Dr. Daniel Kessler's group from Munich, Germany, present their work on functional connectivity measures. And we welcome collaborations in various forms. For example, if you wish to share your work on any topic within the neuroimaging community, please reach out to us. We would be very delighted to host you as a guest. Before Shalaila's presentation, I would like to spend like 3 minutes to have a brief recap of our last event during which our PhD student Yu Dongyu and myself have presented our CentileBrain models. We now have a YouTube channel, and we have the video from the last event uploaded to the channel. If you want to know more details about our CentileBrain model, please scan this QR code or search Translational Neuroimaging Educational Program in YouTube. So first of all, I would like to mention that this CentileBrain model is a team effort of the Enigma Lifespan Working Group, and we would like to express our gratitude to every team member who contributed to make this project feasible. In this CentileBrain project, we have developed sex-specific normative models for 3 brain morphometric measures. These regional measures include cortical thickness, surface area, and subcortical volume. Specifically, we have evaluated different algorithms to build a normative model, and we recommend an optimal algorithm. Then we evaluate the currently available covariant to enhance the performance of the normative models. We also did several sensitivity analysis about the different age bands and the sample size to demonstrate the robustness of our CentileBrain models. Then we evaluated the consistency of the CentileBrain models performance on longitudinal samples. We also provided evidence that the CentileBrain models generalize to diverse ethno-racial group, and also provide evidence of the relevance of the CentileBrain models to mental illness. Then our PhD student, Yu Dongyu, presented our CentileBrain website by showing how to use our online calculation tools and how to use the scripts to design your own normative models with our protocol. This is the recap of our last event about the CentileBrain model. Again, if you want to know more details, please search this program on YouTube, and you will see the videos. Today, we are very excited to have Dr. Shalaila Haas to present her latest work on using CentileBrain models to investigate individuals at clinical high risk for psychosis. Shalaila is currently an instructor at the Department of Psychiatry of the Icahn School of Medicine at Mount Sinai, New York City. Her current research focuses on using machine learning methods to link behavioral and neuroimaging signatures, with a particular focus on psychosis spectrum disorders. Her ultimate goal is to understand the underlying mechanisms driving heterogeneity of the clinical presentation and the course of mental illness, and its relation to accelerated aging and risk factors in vulnerable populations. In her presentation today, Shalaila will cover 3 points of her latest work using CentileBrain. Firstly, she will show us how to apply the CentileBrain models to clinical samples to quantify deviations from normative ranges of neuroanatomical variation. Secondly, she will show us how to compute the summary matrix of these deviation scores. And lastly, but importantly, how to link these deviations with behavioral and psychopathological phenotypes. Now let's welcome Dr. Shalaila for her presentation. Shalaila, I'm passing the microphone to you.
Thank you very much for the kind introduction, and thank you to everyone who's joining today. Yeah, as Dr. Guy mentioned, we're really excited about these different events. We welcome any of the work that you're doing for future talks. Just get in touch with us and yeah, continue to see what it is that we're up to. So let me just share my screen. Okay, can everyone see my screen okay? Yeah, okay, great. Then let's get started. So today, again, I'm Shalaila Haas. I'm an instructor at the Department of Psychiatry at the Icahn School of Medicine at Mount Sinai. And today I would like to share our work on normative modeling of brain morphometry in clinical high risk for psychosis individuals. This was a joint effort between the Enigma Lifespan and Enigma Clinical High Risk for Psychosis working groups, and the first application of the normative models Dr. Guy developed as described in his previous talk. And I have no disclosures. Just a quick outline of the talk today. So I'll begin by providing you with some background information and then describing the aims of the study. Then I will go into the methods where I will present several demonstrations to show you how you can compute average deviation scores, quantify extreme deviations at the level of the individual and for each region, show you how you can compare extreme deviations in your patient sample with healthy individuals, and show you how you can assess associations between global and regional deviations with behavioral measures such as positive symptoms and IQ, which was done within the scope of this particular study. Then finally, I will show you the results in our case example in individuals at clinical high risk for psychosis based on data from the Enigma Clinical High Risk for Psychosis Working Group, and provide our conclusions. So let's begin with some background information. So why would we want to use normative modeling in a sample of individuals at clinical high risk for psychosis? The lack of robust neuroanatomical markers of psychosis risk have traditionally been attributed to heterogeneity within the clinical sample. However, a complementary hypothesis could also be that the variation in neuroanatomical measures of psychosis risk may actually be nested within normal variation in healthy individuals, so that the variation lies along a continuum with healthy individuals, as you see here. One way of tapping into this heterogeneity is through normative modeling. Normative modeling provides a method for studying individual differences and parsing heterogeneity across cohorts. This is possible since this method provides statistical inferences at the individual level by identifying the degree to which each individual, for example, the individual that you see here in red, deviates from the normative pattern of a reference sample. Normative modeling is similar to the use of growth charts in pediatric medicine, except the conventional response variables such as height or weight are substituted for quantitative biological measures such as brain volume, cortical thickness, or surface area. Commonly included covariates such as age and sex can also be substituted for clinically relevant variables. There are multiple different algorithms available for running normative modeling. And in the previous talk, Dr. Guy shared details on the benchmarking study that was done to identify the chosen algorithm that's available on the CentileBrain platform, which is multivariable fractional polynomial regression, or for short, MFPR. And this was based on having the lowest mean absolute error and lowest computational time. I won't spend too much time on how these models were generated, as this was largely covered in the last talk and is available online for your reference on the Translational Neuroimaging YouTube channel. This talk instead will focus more on the application of such models to a clinical sample and some of the different ways the outputs from the CentileBrain model and platform can be summarized to better understand deviations in brain morphometry in clinical samples, such as those that are at risk for psychosis. Okay. Much of this work was largely inspired by that of Jinglei Liv and Andrew Zaleski and colleagues, who used normative modeling to investigate heterogeneity of gray and white matter in patients with schizophrenia. In their study, they found that no more than 17 to 18% of patients deviated from the normative range in any region. They also found 79% of patients showed significant infranormal deviations in at least one region, which represents z-scores less than minus 1.96 standard deviations from the mean, whereas supernormal deviations represent z-scores that are greater than 1.96 standard deviations from the mean. But I'll cover more of this a bit later in more detail. The authors concluded that anatomical loci of schizophrenia-related changes are highly heterogeneous across individuals, to the extent that group consensus maps, which you see in the top row here, are not necessarily representative of most individual patients. Infranormal deviations in cortical thickness were most commonly located in temporal and ventromedial prefrontal cortices, as you see highlighted here, although only 10 to 15% of patients showed extreme deviations in these regions. While for supernormal deviations, these were most common in the paracentral lobule. However, again, here it's even less, in less than 3% of patients that had extreme deviations within this region. So it is currently unknown, however, to what degree deviations from healthy neuroanatomical variation in brain morphometry might be present in individuals at clinical high risk for psychosis, and whether these deviations might explain substantial variance in positive symptoms or IQ. This leads me to the aims of the current study, which were to quantify deviations from the normative range of neuroanatomical variation in individuals at clinical high risk for psychosis, using the large representative normative models described by Dr. Guy in the previous talk. And second aim was to evaluate their overlap with healthy variation, and finally to test their association with positive symptoms and IQ. Again, this work would not have been possible without the support of the Enigma Consortium, in particular the Enigma Lifespan and Enigma Clinical High Risk for Psychosis working groups. So now moving on to the methods. So to determine the final sample, we excluded participants with greater than 5% missing MRI data or missing intracranial volume data. We also excluded participants without positive symptom data to have a final sample of 1,340 individuals at clinical high risk for psychosis and 1,237 healthy individuals that were included and aggregated across 29 different sites from the Enigma Clinical High Risk for Psychosis working groups. The analysis pipeline included extraction of brain morphometric measures of cortical thickness, surface area, subcortical volume. That was done locally at each respective site of the Enigma Clinical High Risk for Psychosis Working Group. Then sex-specific Z-score deviations from the normative models were computed for each individual and every region using the CentileBrain platform, the details of which you see here. So here you have basically the Excel templates that you can download from the CentileBrain website and input your own data in order to upload onto the CentileBrain platform to extract specific output measures, which I'll go into in a little bit. Yeah, and then briefly, the CentileBrain model consisted of a sample of 37,407 healthy individuals pooled across 87 cohorts between the ages of 3 to 90 years. And then again, the algorithm multivariable fractional polynomial regression was used within this final model, and this was developed using 5-fold cross-validation, and the sample was divided into training, 80% training and 20% testing subsets. And again, these templates can be downloaded from the CentileBrain website where you can input your own data and run these models on your own data. And for further details on the model itself and the benchmarking study, again, you can refer to the YouTube channel where we have the recording of the first session by Dr. P. So once you have populated the templates separately for males and females and uploaded them to the CentileBrain platform, you get several different outputs. These were also described in the previous talk during the demonstration, but briefly, here you see some examples, including the centiles, which are extracted for cortical thickness, surface area, and subcortical volume. And you basically get the values for different centiles for each particular age that was included within your sample. So this is not subject specific, but specific to the ages available in the data set that you're working with. And then additionally, you get the Z-scores for each region and each individual that is output into separate files, again, for males and females. These output files form the basis for the analyses that were done in this particular study. On the right, we show distributions of Z-scores across all participants, so all 2,577 participants across healthy individuals and individuals at clinical high risk for psychosis for 3 exemplar regions from the outputs of cortical thickness, surface area, and subcortical volume. And the Z-scores or deviation scores that are generated as output on the CentileBrain platform are computed using this equation, basically subtracting the observed volume for your particular individual from the predicted volume at a particular age and dividing by the root mean squared error. In order to identify global deviation across the brain, we additionally computed global measures of deviation by averaging globally across all 150 pre-surfer measures, across all measures of cortical thickness, and across all measures of surface area, and across all measures of subcortical volume, to basically yield 4 different average deviation scores globally and for each of the different measure types. So this leads me to the first demonstration for today, where I will show you the scripts that you can use to generate the average deviation scores for your own data. Across these different measures, whether you're looking at it globally, across measures of cortical thickness, across surface area, or across subcortical volume. All the code that's presented today is available on this GitHub repository. It also includes a template that you can use where you can input your Z-scores that you download from the CentileBrain website after you apply the models to your own data. Exactly. And so in this first step here, basically what I'm just showing is that you are reading in the data. Here it's this template _Z scores .csv file. it's important if you want to use these scripts to use this particular template, because the order of the variables matters for some of the subsequent analyses. Yeah, and here we're just identifying specific measures, like which columns are the free surfer measures, and that's 6 to 155, which gives you a total of 150 free surfer measures. And then here the Z transformed underscore uni is just creating a subset of your data that just has the free surfer metrics. So the Z score values for each region in separate columns, and then for each subject in different rows. And then here we're just identifying which of the rows is the healthy group and which of the rows is the patient group. And the next step from the matrix that we generated with the 150 free surfer measures and columns and 2,577 individual participants in rows, we can now create indices for each measure type. Again, across all free surfer measures, all cortical thickness measures, all surface area measures, and all subcortical volume measures. So here, basically, we're just identifying which of the columns belong to each of these different measures. And as long as you're using the template provided on GitHub, then you don't need to make any changes to the indices for the column numbers. However, I always suggest checking your data. So don't just rely on the scripts, but double check that everything looks correct when you're running it yourself. Yeah, so here we then have the indices for each of the different measures, as I mentioned. If you want to look at other specific groupings of brain measures, such as looking at specific lobes, you can also create indices for this separately. Then in the next step, we aggregate all 4 indices into one variable and create a loop to compute average deviation scores for each measure type and for each individual participant. So in the end, your output is a matrix with 4 columns of average deviation scores for each subject in rows. So you can see that here. And here it's just showing the loop and creating an index for all 4 of these different measures so that it loops through each of the different global metrics and each of the different subjects, which is your ID variable. And then the other nice thing is that you get the output is then you can save it into a CSV file, and you can use these measures for other analyses later down the line, which I'll also show you. So what I showed you previously was just computing the average across your deviation scores, regardless of the sign. However, you can also repeat these analyses taking the absolute value of your deviations, so ignoring the sign. And then you can also do this for all positive deviations and all negative deviations separately. However, the interpretation gets a bit tricky when using positive and negative deviations, as each subject may have different regions comprising the average deviations. Okay. So in terms of the statistical analyses that were included in the study, we first quantified the extreme infranormal or supernormal deviations, which, as you can see here, represents z-scores. So infranormal deviations represent z-scores below minus 1.96, and supernormal deviations represent z-scores above 1.96. And this is following the work that was also done by Liv and Zaleski and colleagues. Yeah, these metrics can be summarized at the individual level. In other words, in a particular participant, how many regions are at extreme positive or supernormal or extreme negative or infranormal deviation. Additionally, this can also be summarized for each region by asking the question for any given region, what proportion of healthy individuals or patients have extreme deviations in either the positive or negative direction. Then the information for each region can be used to statistically compare whether the proportion of individuals with extreme deviations within each group, healthy or clinical high risk for psychosis, differs using the two proportion z-test. And finally, we assess the associations between regional and average deviation scores with positive symptoms and IQ using linear regression. So now I will show you how you can quantify extreme deviations by the individual or region using the scripts that we have online. So for the first, we're looking at the quantification of the percent of patients or healthy individuals with extreme deviations per region. In other words, for a given region, what proportion of healthy individuals versus patients have extreme deviations in either the positive or negative direction? So basically what you get here is for every for every region, the proportion or percentage of individuals within each group. Here on the top, you have the, well, here we're only showing the patients. On the top, we're showing the percentage of subjects that are at clinical high risk for psychosis with extreme supernormal deviations in a particular region. And at the bottom, we're showing the percentage of subjects with extreme negative deviation in a particular region. And again, the ordering of this depends on the way that you have your data set up. But this, I don't show this step here, but we then compile the data into one table, which can be written out and saved, which has the information on which region belongs to which value. And here, just for the purposes of plotting, in future steps, we just assign the negative deviations. We just assign the extreme negative deviations or infranormal deviations a negative value so that we can plot this on the bar graphs. I'll show that later in the results. But of course, these are showing percentages, so it's not actually negative. So that's just something to keep in mind. And here we just have the example for patients, but you can compute the exact same thing in your healthy individuals, and then use this as a basis for comparison between the two groups. Okay. In the next step, I'm showing how to compute the percentage of regions with extreme deviations per individual. In other words, in a particular participant, how many regions are at extreme positive or extreme negative deviation? The output that you get here is one value per participant for the number of regions with extreme infranormal or supernormal deviation across all measures of cortical thickness, all surface area measures, or all subcortical volume measures. And again, this is aggregated into one table that is written out into a CSV file. Once you have this information, you can do things like compute the percent of subjects with at least one positive or negative extreme deviation for each of these columns. So you can just, basically compute to see what percentage of subjects have at least one extreme deviation per group. In the next step, the information that was computed for each region can be used to statistically compare whether the proportion of individuals with extreme deviations within each group differs using the two proportion z-test in R. And the code to run these analyses is also available on GitHub. Much of this code was supported by Dr. Maria Jalbrzykowski, who is one of the co-leads of the Enigma Clinical High Risk for Psychosis Working Group. So thank you to Maria. But here, basically, you just have a list of some of the dependencies. And in the first step, it's basically just assigning for each particular subject and each region categorically whether a region is infranormal, normal, or supernormal. And normal in this case is just a z-score between minus 1.96 to 1.96. So within the norm, not outside of at the extremes. So you basically get a table, as you see here, where again, every column is a region and every row is a subject. Then once you have this information, you can loop through the regions to compute the two proportion z-test to compare whether the proportion of subjects with infranormal deviations differs in healthy and patient groups. You get separate outputs for the comparison of infranormal and supernormal deviations, as you see here. But yeah, here we just have an example within subcortical volume, which is a little bit easier to depict since it only includes 14 regions, but this is the code for this is also available for cortical thickness and surface area online. Yeah, here you just have the loop that goes through and computes the two proportion z-test using prop.test function in R. Yeah, so the first is showing the infranormal deviations and how to compute that, and the second is showing the supernormal deviations. And this is basically what the output looks like that you can save and refer to later for your own analyses and outputs. And then one thing to note, here we also have the proportions, but since they're proportions, you just have to multiply by 100 in order to get the percentages within each group. Finally, within the clinical high risk for psychosis group, we used linear regression models implemented with the LM function in R to assess associations between positive symptoms and IQ, with each regional deviation and each average deviation score. Age was included as a variable in all regression models due to its significant association with positive symptoms and IQ in our particular sample. So in order to investigate the links between your deviation scores and behavioral measures, such as IQ or positive symptoms, what you'll first need to do is basically add your behavioral measures to the end of the templates _ z-scores.csv file. It's important to add this at the end just to make sure that the column naming isn't, there aren't any issues with the column numbers, because these are referred to for doing the different analyses and different steps. So just be sure to add it at the end. Yeah, for this study in the main model, we investigated the effect of positive symptoms on deviations in regional morphometric brain measures while covarying for age. And here we can see where we loop through each region. So NVAR just defines which columns in the data are the free surfer measures, and it basically computes this function that you see here, linking the positive symptoms with each measure within each regional deviation score while controlling for age. And what you get is basically this output, which gives you the various different results. I additionally also added another line to add the FDR corrected p-values. But yeah, these are the main outputs that you can then write into a table and save. You may want to rename the output based on whatever behavioral measures you are particularly interested in for your analyses, and also renaming the last bit according to your sample or project that you're working on. Then here I'm just showing another example where the only thing that changes is replacing positive symptoms with IQ. Again, you get the results here for the association between regional deviation scores and IQ across clinical high risk for psychosis individuals. If you have IQ data for healthy subjects, you can also repeat the same analysis in your healthy sample. And if you remember from before, I showed you how to compute the average deviation scores. So globally for subcortical volume, cortical thickness, and surface area. Another thing that you can do here is, again, use the outputs that you generated to link the average deviation scores with your behavioral measures instead of looking at each region separately or in addition to, as was the case here. And doing that, we have a separate section within the script where we're basically reading in that average deviation score CSV file. You'll again have to add your behavioral measures to the end of this sheet. And you want to make sure that your behavioral data, of course, is in the same order as your Z-score data. Yeah, and then again, loops through. In this case, it loops through the average deviation scores instead of each region separately. And your output here, you have your average deviation score across all measures, all subcortical volume, all cortical thickness, all surface area, linked with, in this case, IQ. So now moving on to the results that we found in our particular study in clinical high risk for psychosis individuals. So for the first set of analyses, we found similar percentages of clinical high risk for psychosis individuals and healthy individuals with at least one region showing extreme deviation. That's what we have in this table here. So you can see always between the slash on the left, you have the clinical high risk for psychosis individuals. And on the right, you have the value for healthy individuals. And what's pretty striking here is that the numbers are pretty similar, whether you're looking at infranormal or supernormal deviations, and whether you're looking at cortical thickness, surface area, or subcortical volume. Then on the right, you can see that we also found very similar patterns for the proportion of subjects with any given number of extreme deviations, with the majority of subjects not having more than 4 regions that deviated at extremes. So you can see most of the proportion, most subjects fall within having less than 4 regions with extreme deviations across the entire brain. Yeah. And basically, these findings suggest that there's no difference in the proportion of subjects with deviant regional values. Yeah. Looking at this another way, here we report for any given region the proportion of individuals at clinical high risk for psychosis, those who later converted, which is the CHR PC, and then healthy individuals with extreme deviations. So what you see on the left always is the percentage with infranormal deviations, and on the right, the percentage with supernormal deviations, and each of the colors represents one of the different groups. What's striking here is that looking at any of the different regions, there aren't more than 12% of individuals that are at clinical high risk for psychosis or those who later converted that exceeds 12%. So it's a small percentage of individuals with extreme deviations. And you can also show the same thing for the average deviation scores that I showed you how to compute earlier. And these findings suggest that extreme deviations were present in a minority of clinical high risk for psychosis individuals, and that proportions are largely similar to that observed in healthy individuals. The only regions that showed significant differences using the 2 proportion Z test are those highlighted in red. And for the inferior temporal region of cortical thickness, this was significantly different between clinical high risk for psychosis individuals that later converted versus healthy individuals. And for the Banks superior temporal sulcus, this was significantly different only between clinical high risk for psychosis converters and those that did not convert. Yeah. So yeah, as I mentioned, extreme deviations were present only in a minority of clinical high risk for psychosis individuals and at proportions largely similar to those observed in healthy individuals. Looking at the distributions of deviations across regional and average deviation scores in these various different measures for the different groups, whether you're looking at it at each specific region or if you're looking at it from the average deviation score perspective or at specific regions separately, what you basically find is that there's a large degree of overlap between the distributions in healthy individuals and those that are at risk for psychosis. Here in this particular example, we use the hippocampus as the exemplar region because it had the greatest Cohen's d effect size difference between individuals at clinical high risk for psychosis and healthy individuals in this particular sample. But even then, you still see substantial overlap in the deviation scores. So regional and global variation and macro scale measures of brain morphometry in clinical high risk for psychosis is largely nested within normal variation. We also have a script on GitHub for generating the distribution plots yourself for your own data using the same template _z scores CSV file that we uploaded, where you can add your data. There's an accompanying tutorial for the steps of generating such figures that can be adapted based on your own needs. And these scripts were largely supported by Dr. Nicole Sanford from UBC, University of British Columbia. Okay. The results of the linear regression analyses showed significant associations between regional and average deviation scores in positive symptoms and IQ, with higher average deviation scores in surface area being associated with lower symptoms and higher IQ. Global average deviation score was also positively associated with IQ. These findings are consistent with studies in healthy individuals, showing greater cortical surface area being linked, being identified in individuals with higher IQ. At the regional level, greater positive regional deviations in subcortical volume of the left caudate and surface area of the left cuneus were also associated with higher IQ. The caudate has also previously been linked to IQ in healthy subjects, and in this particular study, there were no other subcortical structures that were independently associated with IQ. Again, this was in healthy individuals. In addition to the linear regression analyses, we also used a simple linear support vector classifier with either raw or normative deviation scores. And what we found here was that it revealed near chance level performance in identifying individuals at clinical high risk for psychosis. So the classifier was just predicting whether an individual was healthy or an individual with clinical high risk for psychosis. And here you see the values, very close to chance level. The findings were also similar when looking at distinguishing those that later converted to psychosis versus healthy individuals, the results of which you can see in the paper itself. And again, the code to run these analyses are openly available on GitHub by Sage Rutherford, which is linked here if you're interested in applying these to your own data. And these findings together suggest that neither normative nor raw data were predictive of group status, positive symptoms, or IQ in those that are at clinical high risk for psychosis. So this leads me to the conclusions for the current study. The anatomical loci of psychosis risk-related changes are highly heterogeneous across individuals. Regional variation in the neuroanatomy of clinical high risk for psychosis individuals was nested within normal variation, and the degree of neuroanatomical normative deviations showed minimal associations with positive symptoms, IQ, and conversion status. Some aims for future studies include using individual level symptom or cognitive domains that might be more informative and contribute to deviations in specialized brain regions. Here we only had access to positive symptoms. One of the reasons being within the clinical high risk for psychosis cohorts, we had 2 different clinical batteries that were used to identify symptoms within this sample. So we were limited to the positive symptom domain. Longitudinal changes in brain morphometry may be more informative as psychosis risk markers. And multimodal neuroimaging studies may provide a more comprehensive understanding of the underlying mechanisms associated with psychosis risk. Yeah. So that leads me to the end of the talk. I hope that the various different methods and metrics that were used within this study can be useful to some of the projects that you might be working on. I'm happy to discuss any open questions or yeah, take any comments from everyone here. Thank you.